Yes.
Uh-huh. Okay, great. All right, thank you so much for inviting me here. Let's just get started. Like Sarah said, my lab mostly focuses on understanding how biological interactions and especially how symbiosis impact organismal evolution. So today I'm gonna tell you a few stories about these solar-powered animals. In other words, the animals that perform photosymbiosis. So, photosymbiosis is a ecological interaction between heterotrophic hosts and photosynthetic symbionts. So, in these associations, the symbionts are performing photosynthesis, and then they will provide organic sugar to the host, and the host provides inorganic nutrients and shelters in return. So this is a mutually beneficial interaction. And this association has independently evolved many times in different eukaryotic host lineages, including animals, of course. And within animals, one of the most famous examples are probably corals, stony corals, right? Because corals have association with their algosymbionts, and the algosymbionts can perform photosynthesis. So that allow coral animals to live in these very nutrient-poor oligotrophic marine environments and actually flourish and build massive reefs, allowing them to support biodiversity. So this photosymbiotic interaction is obviously ecologically very important. And then therefore it's interesting or important to understand molecular mechanisms, adaptations, or consequences behind these associations. So we can better understand the future evolutionary trajectory of these associations and perhaps understand their conservation implications. And indeed, in the past decade or so, lots of research has been focused on understanding molecular mechanisms behind symbiosis, photosymbiosis, but mostly focused on cnidarians because of their conservation value. So, for example, in this classic paper, Barrett et al. 2014, they've discovered a particular proton pump, this VHA protein, is associated with the symbiont containing structure in corals. And they're actually pumping a lot of protons into the symbiont surrounding environment, lowering the pH for the symbionts and help increase CO2 concentration around the algae and facilitate photosynthesis. Right, so this is an interesting molecular mechanism for the host to help the symbionts to get more CO2 around their environment. And subsequently, similar mechanisms has been found in both giant clams and photosymbiotic anemones. So it's not very surprising that corals and anemones might share similar molecular mechanisms. They're both cnidarians, relatively closely related in animals. However, for giant clams, now this is a mollusk and it is distantly related to cnidarians. For them to use similar mechanisms to host their algae, this begs a question, for me at least. So, do different animal hosts that have this similar photosymbiosis ecology actually share similar underlying molecular mechanisms, or do they actually evolve their own unique adaptations to interact with their symbionts? Right? To answer this question, we really need a lot of information from different diverse animal hosts so we can do comparative work. And since in the past decades or so, we have a lot more work focused on cnidarians, I want to use a different system, understand molecular mechanisms behind it, and provide more data and information for comparative work. And for that, you heard Sarah says my study system are mostly mollusks, so I pick bivalves as my study system. So within bivalve or clams, mollusks, there are two lineages that are obligate photosymbiotic, and they both belong to the bivalve family Cardidae or cuccos, that's the common name, and they form photosymbiosis with different algal strains or genera within the same family here, Symbiodiniceae, which is also the family of symbionts that corals and cnidarians are associated with. So within Cardiidae, there are two subfamilies that has evolved photosymbiosis. One is Tridactynae, the giant clams. The other is Phrygiinae, or heart cockles. So giant clams are perhaps more familiar to us. They're famous for their colorful mantles, very heavy and sometimes huge shells, and they host their symbionts extracellularly in their mantle tissues. And you can see here, and usually in the in situ, they expose their mantle tissues under the sun so their symbionts can get enough sunlight. So here is a Tridacnogyrus. This is the biggest living clam species on Earth. This is the Great Barrier Reef. You can see the mantles are brown and golden. Those are the colors of the algae. And just to give you a sense of scale of how big this clam is, that's my daughter. She's swimming above the giant clam. You can see she's pretty much as big as the clam, or the clam is as big as her. So that's giant clams. And then our second group are Phrygidae or hard cockles, perhaps less familiar. There are many more genera in this subfamily. And these animals have very different morphological or behavioral adaptations to photosymbiosis. They're not huge, they're small, few centimeters mostly, and they like to bury in the sediment, as you can see here. And instead of opening entirely their shell, many of the species actually have a pretty small gap right here, and a lot of them are using actual shell microstructures to direct light underneath their shells and actually perhaps concentrate or filter or diffuse light for their algal to perform photosynthesis, right? And if you open it up, you can see that the gill tissue, which is here, and the mantle tissue of hard cockles are packed with symbionts. Again, the golden color you see are the color of the algae. And the foot is usually symbiont-free, at least most of the part. Okay? So in order to understand molecular mechanisms and consequences of photosymbiosis in these groups, I'm going to use three different approaches. First, at the transcriptome level, to look at gene expression patterns, and then we're going to dive into the nuclear genome, and then lastly, in the mitogenome. So as a lot of us know, it's difficult to work on non-model organisms if you want to really understand molecular functions. For giant clams or hard cockles, I can't really just knock out their genes yet to understand their function. But luckily, we have a comparative system that allow us to ask comparative questions. So here's a phylogeny of Cardidae, so the cockle family. And you can see that the red lineages are photosymbiotic. Here's our Tridacna, here's some hard cockles, and the black lineages are non-symbiotic. You will notice that in the subfamily Phrygidae here, we have two lineages. One is exclusively photosymbiotic, the other is not. So because we have this sister group relationship within the same subfamily, we can actually look at gene expressions of a symbiotic species and a non-symbiotic species, looking at their differences and maybe infer some potential functions that are contributing to photosymbiosis. And that's what we did. So we picked a photosymbiotic species and a non-symbiotic species here, and then subjected them to some field experiments. So we put both species in three different treatments. Ambient light is the normal light that these animals will usually face in their shallow sandy habitats. Dim light is we're giving them half of the light they usually get. And then dark, so complete dark, we treat them for 3 days. After this, we collected 2 tissue types from both species. For our symbiotic species, if you remember, the mantle tissue is where the symbionts are, and the foot is symbiont-free. And then we also collected mantle and foot of the non-symbiotic species, which shouldn't have any algal individuals in there at all. And then we look at their gene expression patterns and transcriptome analysis. So if a gene in our criteria is involved in photosymbiosis, we want this gene to meet 3 different requirements. So first of all, this gene should express or be highly expressed under normal light conditions in the mantle tissue of the host, because that's where the symbionts live. And second, it shouldn't be highly expressed in the foot because that's symbiont-free. And then, of course, it should not be expressed in the mantle or foot tissue of the non-symbiotic species. But if it is expressed for some other function, right, we express a different pattern. So for the symbiotic species, we expect these genes to be expressed following a light gradient. In other words, in highlight, normal light, where symbiosis is normal, it should be highly expressed. And then in complete dark, when photosymbiosis is shut down, it should be lowly expressed. And in the non-symbiotic species, we do not expect such pattern in reaction to light gradients. Right? So we look at the transcriptome of our mantle and foot of the two different species and then pass all of them through the selecting criteria. Do we find any candidates? The answer is yes, we find candidate genes. So for example, this is carbonic anhydrous, and you can see this is expression pattern in our Phragm suiziens, which is a symbiotic species. In the mantle tissue, we have this nice light gradient following ambient, dim, and dark light. And then in the foot, this gene is not expressed at all. We also did not detect gene expression in the foot and mantle of our non-symbiotic species. And turns out, independently, this gene has been identified again in corals and giant clams as a key gene that is facilitating photosynthesis. So this is enzyme that catalyze the conversion between CO2 and bicarbonates. So these are expressed again around the symbiont containing tissue and cells in these different hosts, and they are again increasing CO2 concentration around the algae and allow them to perform photosynthesis. So it's nice to see that our independent approach landed on the same molecular mechanism that are used by other animal hosts to help their symbionts. Right? And here's another example. This is the Niemann-Pick2 C2 gene, and it's a cholesterol transporter. Again, we have this nice light gradients here, and it's not expressed in the foot or non-symbiotic species. And we know the function of this gene because it's, again, heavily studied in humans. So in human, this is also a cholesterol transporter to transport cholesterol out of the lysosome, right? And if you have mutations in this gene, you have horrible disease. But interestingly, again, it's been studied in corals and anemones, and these researchers found a non-canonical version of this gene is actually transporting symbiont-derived steroids out of the symbiont cells into the host. So from a lot of these candidate genes, we can find that different photosymbiotic hosts actually did not reinvent the wheel. They a lot of times don't use a novel gene or a novel mechanism to help them interact with their symbionts. And instead, they actually utilized certain mechanisms that perhaps already evolved as the common ancestor of animals and repurpose it for their own use and for their own interaction with symbionts in terms of metabolic exchange and facilitating photosynthesis. And these are not the only two genes that we found that are shared by a lot of different lineages of hosts. Actually, all the things I'm listing here are genes that fits our criteria, but also has been studied in other animal hosts, indicating they're facilitating photosynthesis and symbiosis. And they involve, immune responses to maintain their symbiont cells in the host body, photoreceptors, reactive oxygen species management, nutrient transportation, metabolism, etc. So in other words, we found lots and lots of genes are actually shared among distantly related hosts, which is very interesting. So do bivalves actually have any of their own unique mechanisms when interacting with the symbionts, right? Because they do have different mechanisms of hosting symbionts. For example, in cnidarians, the symbionts are hosts within their cells, but in bivalves, they're extracellular. They have a tubular system that's hosting symbionts. Does any molecular mechanisms are involved in these interesting interactions? And the answer is yes. So we've looked at, for example, upregulated genes and gene ontology terms in our normal functioning photosymbiosis compared to dark conditions, compared to tissues that do not have photosymbiosis. And what we found is a lot of these functions are related to cilium, microtubule, and all of those, right? So it's quite interesting in our histological sections, we do see that on the right, the symbiont-containing tissues here are actually showing the existence of cilium and cilia around here. And also interestingly, in the older study looking at the histology and morphology of heart cockles, we see that in both the longitudinal sections and cross sections of the symbiont containing tubes, long cilia has been shown in all these structures. And the author also indicated that actually in the kidney of the animal host, he observed half-digested symbionts in there and potentially being expelled out of the host. So combining all these histological evidence and our molecular function, it seems to indicate that these long cilia in the tubules are used to mobilize the symbionts and actually move them from different places in the host to other destinations, potentially as a mechanism for symbiont population control or moving them into more light friendly environments. So it's really good to see that molecular work and histological work actually converge to similar mechanisms of how photosymbiosis is working in our bivalve system. So to summarize this part, right, we found both shared and novel molecular pathways that are potentially involved in bivalve photosymbiosis. But from this work, we're only looking at expressed genes or kind of only scratching the surface of this vast complex molecular pathway. So in order to really look deep into some of the consequences of photosymbiosis, we do need to dive into the actual genomes of these organisms and look at their genomic structures. So thanks to the Aquatic Symbiosis Genomics Project, funded by the Welch Sanger Institute and the Moore Foundation, and also our own work with dovetails, we now have 11 photosymbiotic bivalve genomes assembled at the chromosome level, including different species of heart cockles and giant clams as well. And we haven't really processed all 11 bivalve genomes, but today I can show you some of our findings in 3 giant clam species. So Tridacna maxima, Tridacna corosea, and Tridacna gigas, and this is work done with Sarah together. So first, with these genomes, we can start to look at gene family evolution and gene evolution in these different groups. So here I'm showing you a heat map of gene family evolution and comparing giant clams, these 3 species, with 13 other different mollusks spanning from other bivalves and gastropods as well. So each column is showing a species, a mollusk species, and each row here is an exemplar genes, has certain functions that we annotated. And then each numbers in these squares indicating how many genes are found in a particular gene family in this particular animal's genome. So here is a heat map showing what we call gene family expansion or taxon-specific genes. Right? So you can see in all our 3 giant clam species, they either have many copies of genes belong to this gene family, or at least that gene is present in their genome. However, if you look at the rest of mollusks that we used, they really don't have any genes in their genome that belong to all these gene families. And if you look at the function of these gene families, they're involved in immune regulation, involved in calcification and growth, reactive oxygen species interaction, and metabolism and nutrient transportation. So again, similar functions as we detected in our transcriptome study, which makes sense. It's involved in hosting a large population of symbionts in your tissue, so you need a certain level of immune adjustment for that. And then for giant clams particular, their heavy shells and their big sizes are potentially being contributed by photosymbiosis and light-mediated calcification. And because they're under a lot of oxygen stress constantly asserted by their photosymbionts, they do need molecular mechanisms to handle these reactive oxygen species, and of course, carbon and nitrogen metabolism between these organisms. So it's good from this genomic analysis, we do see signatures of photosymbiosis. What about genes that are missing then in giant clams but found in other groups? So here's the opposite of gene family expansion is our contraction study. Again, you can see from this analysis, a lot of genes belong to these gene families are present in all the rest of the mollusk species, but not in our Tridacna, all 3 of them at all, right? And if you look at the function of these gene families, you can see overwhelming group of immune-related functions that are potentially lost in Tridacna, and then some other metabolic important transporters. So it's interesting to see both immune-related genes being expanded and contracted in giant clams. Why is that? Well, for now, our thought is that for a giant clam to be successful in photosymbiosis, it actually requires a pretty balanced trade-off in their immune system. right? So on one hand, you need to actually recognize your particular strain of symbionts, allow them to enter your body, allow them to flourish. On the other hand, you need to distinguish the rest of the oligosymbionts and be able to reject them. But also, in order to host high density of basically foreign species living in your body, you also need your immune system to be kind of tamed and not overacting, attacking your symbionts, right? So we think that's why we see both expansion and contraction of very important immune-related pathways. For example, we see expansion of some novel pattern receptors, which are used to recognize symbionts or pathogens in the marine environments, right? So it makes sense that they have novel receptors to recognize their own symbionts. On the other hand, we also see contractions of common recognition patterns that are used by other metazoan animals to recognize pathogens and other microbes in the environment, because you don't want your immune system to be constantly alerted and hyped up to recognize all the other microbes, right? You want to keep your immune system function relatively tame and not over responding so the symbionts are not attacked. So in other words, we think photosymbiotic hosts must have a pretty unique and different immune system, and the regulation might be different from non-symbiotic hosts. So these are our immune system related patterns. And then we also notice that for metabolic and growth, and again, here's a subsample of the genes you see here, a lot of the genes in this C1q tumor necrosis factor family are completely missing in our giant clams, but they are pretty abundant in other mollusks. And again, we're lucky because this gene is well studied in humans. Otherwise, we wouldn't really know their functions much, right? But in humans, and this gene is used to actually regulate glucose level in your body. So it's heavily studied in diabetes researches. And it's even been knocked out in mice systems and see what function it plays when this is knocked out. And it turns out when this gene is knocked out, mice actually stop regulating their food intake and they become and have a really high appetite. And they start to keep eating and increase their body weight. So in other words, they became giant mice instead, right? So I can't say this gene is exactly involved in photosymbiosis in giant clams, but from this function, we can infer that, in photosymbiotic systems, the sugar in the blood or the hemophil of the giant clams is being supplied symbiotic derived carbon or sugar, basically. So if your system is constantly being supported high level of sugar from the symbionts, it might make sense to not have these mechanisms to constantly detect glucose in your body and constantly trying to regulate its level, right? It would not be beneficial for photosymbiosis. And that might be a reason or selection pressure for giant clams, all three species, to start to lose this gene and eventually completely lost this mechanism in their metabolic regulation. So all the things I talked to you before is really focused on protein coding genes and other gene expressions. And that is only really a small percentage, as we know, of animal genomes. And animal genomes has other genetic elements that are beyond genes. So if we zoom out and look at the entire genomic structure, we also find interesting patterns in photosymbiotic animals. So for example, here is Tritanomaxima. If we're looking at their genomic composition, you can see that the genes are only about 30% of their genome, and the rest, 70% of their genomes, are composed of repetitive elements. And this is interesting. So we look at other photosymbiotic animals and see if they have similar patterns. And it turns out we found similar patterns in Tritanochorosia, 69%, and Tridacna gigas, 67%. We also dig into literature looking at other symbiotic systems, and we found that even in anemone squirrels or chemosymbiotic bivalves, their genome is reported to have higher level of repetitive elements compared to their non-symbiotic relatives, right? And a large percent of their repetitive elements here are actually transposable elements. So this makes us wonder, why do we consistently find this pattern across diverse different hosts? Is it because these hosts somehow are end up with higher level of transposable elements because of their interaction with their symbionts, or is it simply because they have larger genomes, or this is more random effect? Right, so we start to look at the relationship between genome size and the total transposable elements interactions. So as you can see, here are plots of other mollusks, including bivalves that are labeled here. You can see a pretty much tight correlation between genome size versus transposable element size. The bigger your genome is, the more transposable elements you have. And it's pretty linear right here. However, if you plot our giant clam data on this graph, you can see that they are really outliers. Their genomes are not particularly big compared to other animals. However, their transposable elements percentage is much higher. So this is not really an effect of genome size, but really showing that giant clams is having higher or unexpected level of transposable elements in their genome. And why is that? So to further look into the relationship between symbiosis and transposable elements, we estimated the insertion time of different transposable element groups in our Tridecna genomes. So here is a graph showing on the axis is time. So 0 is current, 120 million years ago. And then on the y-axis is the number of or the size of transposable elements inserted at that time frame. And within this time frame, you can see the green bar here around 30 million years ago, and that is when photosymbiosis first evolved in giant clam. And here we're showing you this data for 4 different species, Tridecna maxima, Gigas, Corosia, these are the blue lines. And then we also have a non-symbiotic cockle, cardiates here, showing in red. And through this pattern, you can see within this 30 million time frame, we see a sharp increase of transposable element size in all 3 Tridecna species, but you don't see that pattern in our non-symbiotic cardiates here. So with this timing, it gives us more evidence that at least the timing of photosymbiosis evolution and the increase of transposable element size are associated here. Right, so it indicates that there might be potential relationships between the more than expected large size of transposable elements and being photosymbiotic. So what are some of our hypotheses, right? The first thing to think about is, yes, as a host, you're living tightly, closely with a foreign species for the rest of your life. So between symbionts here and our giant clam, it's possible that transposable elements can be transferred from their symbionts to their host, therefore increase their size of genomes being inserted with different transposable elements. But secondly, if we go back to look at our genomic data and transcriptome data, we do constantly observe this phenomenon of suppressed immune system. Like I said before, the host needs a relatively downregulated immune system to work with their symbionts. And because transposable elements has viral origin, it is possible that 30 million years ago or even earlier, these suppressed immune interactions are making their host more vulnerable to viral infection and also more vulnerable to transposable elements insertion. And in addition, if your immune system is damped, you actually are potentially losing several mechanisms to control transposable elements replication within your genome once they're inserted. So this might also contribute to the large size of TEs because first of all, you're more vulnerable to their insertion. And second of all, once they're inserted, you might have less chance of actually regulating them and actually control the replication in your genome. Okay, so from this work, we can see that photosymbiosis likely influenced both the gene evolution in these animals as well as its genomic architecture evolution. And let's not forget that animals also have a different sets of genomes. In addition to nuclear genomes, they have mitogenomes. So do we find similar patterns between the nuclear genomes and the mitogenomes? Well, usually animal mitogenomes are considered pretty conserved. They have these 37 genes coding proteins and ribosome RNAs for respiration functions, right? So here I'm showing you 2 mitochondria genomes. One is human, one is sea snake. They pretty much look identical here. Okay? So what happened to our photosymbiotic system then? do they also have pretty conserved mitogenomes that not showing any signature of influence by photosymbiosis? So this time we have able to assemble mitogenomes of a lot of different photosymbiotic species, including hard cockles and tridecna. And we also have mitogenomes of non-symbiotic cardioids here. So then we can take a look and see whether their mitogenomes are also being inserted a lot of repetitive elements and potentially TEs. So how do we look at that? Here is an example. So theoretically, if your mitogenome has no repetition, like this freshwater snail here, and you plot it against yourself, you should observe a straight line here, right? Because every nucleotide sequence should match itself and there's no repeats. It should be a pretty clean straight line. And theoretically, if you have repeats, lots of repeats in your mitogenome, you should see patterns like this because, this chunk of the sequence is matching itself, but also matching a lot of other parts of the mitogenome. Similarly, here, you will find lots of repeats, so one chunk matching to multiple locations in yourself. And that's the pattern you would expect. So we did this plot with our non-photosymbiotic and symbiotic hosts. So this is the results of our non-symbiotic species. You can see that mostly there's no repeats detected except small percentages here and there. Are you ready to see the symbiotic species? So here are our results for our photosymbiotic species, both hard cockles and tridecna. You can see that we detect simple repeats, we detect complex repeats, we detect crazy repeats that I don't even know how to interpret this graph at all. I have no idea. It looks like alien message to me. So the message is that yes, we do see complicated and extensive repeats in our photosymbiotic mitogenomes as well. And a lot of the repeats are actually nested within the so-called non-coding region in the mitogenome. So here again is our phylogeny. I'm showing you different mitochondrial assembly of our photosymbiotic species. You can see the red shaded region are the non-coding region where most of the repeats are found. And sometimes these non-coding regions are almost half of the entire mitogenome. And in contrast, this is our non-symbiotic mitogenomes, and you can see there is a short non-coding region here, and the rest don't really have detectable significant non-coding region. So we do see significant differences between our photosymbiotic mitogenomes and non-photosymbiotic mitogenomes. And then again, this begs the question, what are the potentially functional implication of this? And are those repeats made of transposable elements? So here we can take a look, right? For understanding that, we need to actually annotate genetic elements in these non-coding regions. And that is not always easy because some of the elements might not have references or might not have been discovered in other organisms. So we can't really always annotate everything in this non-coding region. But some interesting things we found is, for example, in fragum, in this non-coding region, we have 11 copies of tRNA S gene, even though this species already have 2 regular copies in the coding region, right? So this again screams repeats to me, and what is bringing these repeats to these regions? And again, transposable elements is one of these mechanisms that creates lots of repeats in the genome. So we, in addition, start to annotate other elements in this non-coding region, and I'm here showing you 4 different examples, 3 hard cockles, 1 tridecna, and the blue region here you see are the non-coding regions. And again, the blues are tandem repeats. You can see similar patterns. They have lots of crazy repeats in this region. In particular, this species, Fragum suiziens, it has a mitochondrial genome size of 92,000 base pair, which is one of the biggest animal mitochondria that have ever been reported. And again, you can see the non-coding region, so C of this group is the majority of its mitochondria. And if we actually zoom in, you can see that a lot of these repeats overlap with known transposable element segments in the genome, right? They're overlapping with all the yellow or orange colored transposed elements. And in addition, not only these are transposable elements or segments of transposable elements, they also fall within reading frames, which is purple. right? The open reading frames largely overlap with these TEs. So in other words, not only the TE and the repeats are there, some of them are potentially even expressed in the mitochondria, whether it has molecular functions or it's just a side effect of them being there and haven't lost their ability to be expressed. We don't really know, but it indicates that for all our photosymbiotic species, their mitochondria is also loaded with transposable elements, just as their nuclear genome. And what are some of the mechanisms that are in kind of resulting in this insertions of transposable elements? Well, first, we thought about, photosymbiotic organisms are again always under expression and pressure of oxygen because their symbionts constantly performing photosynthesis under light. And because mitochondria is important organelle for cell respiration, this high level of oxygen stress might be causing it to have replication errors, for example, and introduce duplications and tandem repeats. And this has been observed in deep sea bivalves and also shrimps living in unusual oxygen conditions. So it has been shown that animals with unusual level of oxygen do have mitochondria that show expansion of their control region and duplication in the non-coding region. So that's one possibility. And of course, the second possibility is that some of these unannotated elements in this non-coding region represent novel genes or RNAs that might play important functions in photosymbiosis mitochondria interaction with their symbionts with the environment. But that will need a lot of further validation to actually test these hypotheses. And of course, the last is similar hypothesis as our nuclear genome interpretation. Maybe this is a side effect of symbiosis. Because of the dampened immune system, it allows a lot of T insertion, and the animal is not able to keep up with the insertion and repetition, therefore resulting in the huge expansion of T in the mitochondrial genome. Right, so a lot of them will need further validation analysis to actually test any of our hypotheses. So in a clamshell, with all the projects I told you about, here's what we found. So first of all, photosymbiosis is an important driver of invertebrate molecular evolution at the gene level, transcriptome level, and the genomic architecture level. And we do find that ancient gene repertoires at the ancestor of metazoans might contribute to our photosymbiosis molecular toolkits. A lot of them might be repurposed by photosymbiotic hosts. And we also found lineage-specific or novel molecular adaptations in these genomes. And lastly, they are potentially genomic consequences of photosymbiosis by introducing transposable elements and repeats in different regions and structures of the genome. So with that, I'd like to thank my students and postdocs who did the work, and of course, wonderful collaborators, Carl and Sarah, and Joe and Lisa for supporting this work and my funding sources. And with that, I'm happy to answer any questions.
Thank you, Jinshun.